during my first visit, and obviously a second visit was cut short. So at least I'll be coming to see you all in July, and I look forward to meeting you distantly and in person. So as Eric mentioned, I've been working on organoid technology, and I'm going to tell you a story today about how I've been making tonsil organoids to understand the cellular and humoral responses to flu viruses.
I'm sorry, Lisa, let me just interrupt to say. So what I meant to mention is, because we're doing this by Zoom, and I'm not sure how many, we have 30 participants already. So unless there's a really pressing question, I would ask all of you to use the chat feature of Zoom to put your question, add your questions in. If you have something that's urgent, please go ahead and interrupt. But otherwise, I would ask you to mute, take yourselves off video until the end of this, and ask questions by chat. Sorry, why don't you go ahead then, Lisa?
Sounds good.
Lisa stays on, but we can all mute.
I'll just talk into the void. All right, so as I mentioned, I'm interested in understanding adaptive immune responses in humans and what's really going on inside those lymphoid tissues. So just as a quick reminder, a lot of the action of the adaptive immune response is happening inside lymph nodes and other lymphoid tissues. And within these tissues, you have these germinal centers, which are the sites where B cells are affinity matured, where somatic hypermutation can occur, and class switching as well. And T cell response is basically honed primarily by two competitive processes that have varying degrees of intensity depending on what model you're looking at and what antigen. And many of these processes have been described in detail, particularly in mice, by people like Shane Crotty and Hai Chi, Gabriel Victoria, Michelle Nussensweig's group, etc. But basically, you can distill this down to two different processes. One is for B cells to compete for access to the antigen itself. And as you can see here, that's proposed to be through decoration of the antigen that's bound to follicular dendritic cells, although this is actually a quite contentious a model, and really there's no direct evidence of this occurring. And the second competitive process, which is a little bit more well-defined, is for access to T cell help, primarily from T follicular helper cells, which are producing things like CD40 ligand stimulation, co-stimulation, and also cytokines like IL-21. So I always like to put up this slide, but given our current circumstances, I think it's becoming much more obvious that it would be much more helpful to have appropriate models of the adaptive immune response that would allow us to accelerate and improve vaccine candidate testing. As I'm sure you're all becoming rapidly aware, testing and developing vaccines is often a slow and laborious process, and it's incredibly expensive because it's largely an empirical science. It's also really difficult to get at the actual cells that are responsible for making a good response. And that's because getting access to lymph nodes or tonsil or other lymphoid tissues in a vaccination type context can be quite difficult. And essentially what that means is that candidate vaccines are going into clinical trials with really not so many opportunities to be able to test what those responses will look like in humans first. And from personal experience, and I'm sure many of you have encountered this as well, measuring things in the blood can be misleading or a poor predictor of protection, especially for vaccines, but also for other treatments as well. And so when I started my postdoc in Mark's lab, I sought to develop a model that would represent the human adaptive immune response in vitro, and that we could have control over and manipulate to try to help bridge this gap between animal model studies and human clinical trials. And this is really emphasized by looking at some of the most difficult pathogens that we're still dealing with in current times. Of course, we could add coronavirus to this list now, but you can see, though many things looked promising in preclinical tests, really that hasn't translated to good effectiveness in people. And so we continue to struggle on these things, and we really don't know what we should be measuring to determine efficacy. So that is where human immunology comes in and systems approaches. So I would say that we've learned a lot from being able to study mouse models. Mouse immunology will always have a place in immunology. It's how we understand at the molecular level most of the time how all of these systems work. But really, we haven't done a great job of translating those findings into understanding human diseases. And so I would propose that we should study the human immune system more directly. This has been a challenge, of course, for all the reasons that I already talked about, but there are new technologies coming online that can serve as alternatives to these traditional type of mouse experiments. And so I'll share with you today what one of my solutions to this problem would be. Okay, so this is where the organoid stuff comes in. So for those of you who don't know, an organoid is an in vitro model that tries to recapitulate various features of the original tissue or organ that it comes from. That's including the function, of course, the cellular composition, and ideally also the structure of the in vivo tissue. And organoids for the last 10 years have been pretty hot in lots of fields. You can see just a few examples here of various tissues that people have turned into in vitro organoid systems. But a key thing that has been missing from many of these tissues is the immune system. So most of the time, immune cells are totally neglected, despite the fact that they play a very critical role in vivo. So I decided that it was time to prepare a lymphoid tissue organoid. And to do that, I started working with tonsils. The reason why so there are roughly 750,000 tonsillectomies performed annually in the US. And as I mentioned, many of these tissues come from healthy people. So what does a tonsil look like? It has some similarities and some differences from a lymph node. As you can see here, tonsils, like lymph nodes, do have germinal centers. As you can see, one example here and one here. But they also have a unique feature, which is that they are directly exposed to the external environment in vivo. And so they have this squamous epithelium, and they also have blind-ended crypts that sort of cover the epithelial surface of the tonsil. And this makes it an ideal situation for antigen uptake, as this is basically a physiologically relevant way for antigens to be taken up in the mouth. So given all of those features, I attempted to build a model that recapitulates the major features of a lymph node's functions to coordinate T and B cell responses in a physiologically relevant way, to identify some mechanistic features that will help us better understand how human adaptive responses are wired, and then also can we test the ability to respond to numerous antigens, ideally including naive antigens, and also how that can be modulated with adjuvants. So as you can imagine from the title of my talk, I chose influenza as a model vaccine. I have a lot of experience working with flu viruses and vaccines from my previous work. And also it's a good model because we know a lot about what flu vaccine responses should look like from actual in vivo immunization studies. And there are lots of formulations that we can test out. So for the rest of this talk, I'll be talking about mostly the live attenuated influenza vaccine. It's also known as flu mist. And essentially it's given as a spray, mostly to children. But there's also the classical inactivated flu vaccine, IIV. That's the intramuscular one. There's an adjuvanted form of that same vaccine called Fluad. And of course, we can compare that to what actual virus responses look like. So this is overall what the organoid workflow looks like. We collect tonsil tissues from Stanford's Children's Hospital. And at Stanford, at least, most of these surgeries are happening for obstructive sleep apnea in both adults and kids. So overall, the tissues that we collect here are quite healthy. We can mechanically dissociate that tissue entirely into a single cell suspension. And then we can actually just cryopreserve those tonsil cells effectively the same way that you would a PBMC sample. And in fact, if you look at the cell composition of tonsils, they're almost entirely lymphocytes. So we can treat them roughly similarly to PBMC samples. When it's time to prepare organoids, we simply thaw those cells, stimulate them with the vaccine or virus of interest, and plate them into a transwell plate. This transwell has a porous bottom, not so porous that cells can go through, but enough that media can exchange. So effectively, we're giving them sufficient nutrients, but we're also keeping the cells at very high density. And also that porous surface is made of Teflon. So it encourages the cells to attach to each other rather than to the bottom of a plate, for example. And after 5 days in culture, here are some examples of what those organoid cultures look like. So in unstimulated cultures, you can see these white dots here. Those represent areas where the cells have reaggregated in vitro from the single cell suspension. And you can see when we stimulate with live attenuated influenza vaccine, LAIV, we get much more structure occurring in these organoids. And in fact, you start to see structural features by about day 2 to 3. Having seen these macro differences in the structures of the organoids, we then went on to characterize various features of the response. So we looked by flow cytometry for evidence of B cells differentiating into plasmablasts. And you can see on day 7 of the response that the flu-stimulated cultures are producing substantially more plasmablasts than unstimulated control cultures from 15 different donors. We can also detect flu-specific antibodies being secreted into the culture supernatant. In this case, we're looking specifically at IgG, but we also see IgM and IgA production. And you can see most donors are producing flu-specific antibodies when we stimulate them with the vaccine, and not when we leave them unstimulated. In a subset of these donors, we looked for evidence of antibody-secreting cells by ELISpot, and indeed you can see that there's actually quite a bit of variation among individuals, but we always see more antibody-secreting cells when we stimulate with LAIV, and almost none in unstimulated control cultures. A feature of these organoids is that we can actually make replicate cultures and harvest them longitudinally to be able to understand how the responses are changing over time and with stimulation. So for example, I'm showing you just one donor here with serial collections on different days, and then assessing the total B cells by phenotype with CD27 and CD38 expression, which broadly help us characterize B cells into different cell subsets, including naive, pre-germinal center, germinal center, plasmablast, and memory phenotypes. You can see in the LAIV stimulated cultures on the top here that we see a slow but steady increase in plasmablasts that roughly corresponds to the kinetics of a response that you would typically see in vivo. Somewhere between day 7 and day 11 is where you start to see a substantial plasmablast response, and that this is much less so in unstimulated cultures. With the same type of kinetics experiments, we can also look at the antibodies being secreted into the culture supernatants. So here I'm just showing you examples of different profiles that you might see from LAIV stimulated in blue versus unstimulated in red cultures from different donors. So the classical type of response that we see is that somewhere between day 5 and day 10, we see substantial increase in antibody being produced in these cultures when we stimulate with LAIV. You can see that some donors are a little bit slow to the party here. They respond a little bit later, but they still make a good response. We see the occasional donor that makes a quite weak or delayed response. And then we see the occasional donor that makes almost no response at all. And actually we
Lisa,
can go.
can I just ask you then,
Sure.
in terms of the variability, is this variability how do you explain the variability? Is there variability in composition of the cells? Do you think for some of these, even though they're healthy individuals, they might have underlying infection or inflammation? What do you think accounts for the variability?
Yeah, so for example, this donor takes daily inhaled corticosteroids. And so there's a possibility that that immune modulation that they've been receiving as treatment for their inflamed tonsils is actually resulting in a poor antibody response. So you can imagine that the clinical history of the donor, their flu exposure history, their age roughly might correlate with that as well. So I imagine we could even break this out by individual strains and see how they respond to the different strains contained within the vaccine as well. Okay, so far I've shown you that the organoid cultures are capable of producing microgram quantities of specific antibody, and that's a good amount for us to be able to detect and analyze. But then the question becomes, are we just eliciting some pre-existing memory cells that exist in the tonsils? Or are we actually able to sustain and stimulate further maturation of a flu response in vitro? Because if all we're doing is expanding a small number of pre-existing memory cells, it's not very interesting or useful. So we did a series of experiments to try to understand what contributions memory versus naive cells may be making to these responses. So the first thing we did was we looked for expression of activation-induced cytidine deaminase, the enzyme that's responsible for double-stranded breaks and allowing somatic hypermutation and class switch to occur. You can see that we measured AID expression just by flow cytometry intracellularly on days 0, 4, and 7, and we looked at different B cell subsets. And we found indeed that germinal center B cells on day 4 have substantial increase in AID expression compared to day 0 tonsil cells. And that this starts to come back down by day 7 once we've seen from other cultures, they're already making an antibody response. Presumably at that point, the funding have already been selected. You can see we see substantially less AID expression in the B cells where you would not expect to find it, such as in naive B cells. Okay, we've also done some imaging studies, which frankly have been very technically challenging, but also very interesting. What we find is we look for light zone and dark zone markers, CD83 and CXCR4 as indicated here with these different colors. And we can find evidence of separation of B cell subsets based on those expressions. So for example, here I've highlighted where we would anticipate a dark zone might be compared to a light zone, and just another example over here where we see separation of those cells. We've also used repertoire sequencing to try to understand how the B cell response is developing over time and under different conditions, and how we might be able to regulate that potentially. So for example, we have a tool where we can actually detect hemagglutinin-specific B cells by simply taking hemagglutinin protein, which is the main antigen for flu, especially for flu vaccines. And that's biotinylated, and then we can come in with a streptavidin secondary against this to be able to detect hemagglutinin-specific B cells. So essentially, we can look for HA-specific B cells on day 0, culture those organoids under different conditions, and then look again on days 7 to 10 to see if we've increased the number of B cells that are specific for HA or also changed their affinity. So the first thing we did was just characterize under normal conditions what kinds of repertoire sequencing, what kind of repertoire do we see from HA-specific B cells. I'm just showing you one example here from single cell BCR sequencing that we did. Where essentially we sorted the hemagglutinin-specific B cells here on day 0, and then we sorted on day 7 in the presence of LAIV stimulation for 7 days. And we assessed the diversity of those B cell receptor sequences. So basically what I'm showing you here are in black dots, the day 0 hemagglutinin specific sequences. So these are essentially the pre-existing B cells. that are specific for this A California hemagglutinin protein. Then we looked again on day 7 and see a few clonal expansions as shown by these open circles here. They also have multiple isotype usage, so we see some examples of IgG, IgM, and IgA. But the overall message here is that the response is quite diverse. As you can see, the V genes being used are quite diverse, and so that suggests that we're not just expanding a small population of pre-existing cells, but that we actually have a diversifying response. So we wanted to push the system even farther and see what would happen if we eliminated any pre-existing memory B cells or non-naive B cells, and also deplete any pre-existing high affinity hemagglutinin specific B cells. So essentially, if we put the system in a condition that would look more like a naive individual, are those cultures able to produce and sustain a flu-specific response? So to do that, we take our thawed tonsil cell mixture. I depleted the pre-existing hemagglutinin-specific B cells, as well as any non-naive B cells based on their phenotype. I prepared cultures from those. And then we repertoire sequence on day 7 to 10. So this is what that looks like essentially. So you can see we've depleted the HA-specific B cells effectively with sorting. By day 10, we see a small but detectable number of hemagglutinin-specific B cells have returned in the cultures. And we did this with 4 different donors, and we can actually see flu-specific antibodies being produced from 3 of the 4 of those donors. So this culture system does appear to be able to sustain a naive type response when we deplete the pre-existing cells. We can also look for evidence of somatic hypermutation and whether we are seeing an increase in mutations over time from these naive cultures. So just to give you a picture of what those cells might look like at the beginning, if we sort on naive B cells with no pre-existing affinity for the A California hemagglutinin, we see pretty much 0 mutations from the germline lineage for, in this case, heavy chain BCR sequencing, as you would expect. If you look at the pre-existing cells of the tonsil that have flu specificity, you can see they have roughly 5 to 10 mutations, but that this varies by donor. If we look at the organoids produced from those naive cultures, which essentially had 0 mutations at the beginning, you can see in unstimulated cultures, they largely remain unmutated, as you would expect. If we look at flu-stimulated cultures, however, we see an increase in mutations. So that suggests to us that the B cells with at least low affinity from the naive pool that were capable of responding to the flu vaccine that we provided are increasing their mutations to gain affinity for LAIV. And if we look at the clone size from these BCR heavy chains, you can see in the LAIV stimulated cultures, we can find clones that are larger and have inferred specificity for the A California hemagglutinin, just based on their sequence and the pre-existing known sequences that we have. The thing that I'm really excited about with this system is that we can actually start to ask mechanistic questions about human adaptive immune responses that we typically can't ask in humans, or we can really only look at in a mouse system. So just a really fundamental question to start with is, what are the cell types that are required to actually allow for an adaptive immune response to happen? So to do this, we can deplete individual cell types to understand what happens when they are missing. And we can also take individual cell types and mix them back together in different combinations and see what happens. So you can imagine there's lots of sorting experiments that are happening here, but I'll just show you a couple of examples. So we've done these depletion experiments with several different cell types, as I'm showing on the x-axis here. I'll just highlight one particular example and walk you through it. So for instance, when we depleted the myeloid cell population, which presumably has some monocytes and some classical dendritic cells, as well as depleting the plasmacytoid DCs. So basically trying to get rid of any antigen-presenting cells. We see that the plasmablast potential of those cultures declines substantially, almost to the point where there are no plasmablasts being produced compared to the wild-type control cultures. You can see the representative flow staining here for that. This also corresponded to a substantial decline in flu-specific antibody production compared to the wild-type control cultures as well. So effectively, without APCs, you don't get a flu response, which makes sense to us from what we know from mouse experiments, of course. We saw similar things when we actually just depleted the plasmacytoid dendritic cells. And we could look back at the literature from Jacques Bancherot's group. In the early 2000s, they showed that actually you need PDCs to produce type 1 interferon to help stimulate B cell differentiation into plasmablasts. So we were able to directly test that by essentially producing plasmacytoid DC depleted cultures and then supplementing them with a cocktail of type 1 interferons. And you can see that when we supplement with interferon, we are actually able to rescue plasmablast differentiation almost to wild type levels in the cultures. And also we're able to rescue flu-specific antibody production. So in this way, we were able to show the essential role of the PDC in this particular culture system, and also from what we know from the literature, is indeed for production of type 1 interferon. I've done a lot of work previously on T cells in flu, and so this is an optimal way for us to be able to look at some different T cell phenotypes and also understand what their contributions are inside the actual tissues in addition to peripherally. So it's nice to come back to T cells again after doing all these B cell experiments. So what is the role of the CD4 T cell in stimulating and sustaining the flu-specific antibody response? We looked duration of the CD4 T cells to deplete them in both unstimulated cultures and flu vaccine stimulated cultures. We looked for plasmablast differentiation. And you can see under wild type conditions, as I showed before, with flu vaccine stimulation, we get more plasmablasts. Under CD4 depleted conditions, it actually doesn't seem to matter whether we have unstimulated or flu stimulated cultures. We see overall a decrease in plasmablast differentiation potential, though there's still a little bit of plasmablast here. What was surprising to us, however, is that even when we depleted the CD4 T cells, a substantial proportion of the cultures were still able to produce some flu-specific IgG. So this was very puzzling to me, but we were able to go back and tease this apart a little bit. So we looked at the clinical features of these patients, and of course an obvious example would be the age of the patient. You can imagine that that might have an effect on the ability of these cultures to respond. So for example, we know that very young children have quite poor T-independent responses. And also, children who are younger are less likely to have a pre-existing response to flu. So we just stratified that previous data based on the age of the patients. So we looked at kids who were 2 to 5 years of age in red, and kids who were 6 years or older in blue. And you can see under wild-type conditions, there's no significant difference in the flu-specific antibody production of very young versus older kids. See, there's maybe a little bit of a trend of increase. However, in very young children, the CD4 depleted cultures were entirely unable to produce flu-specific antibodies. And so that suggests that there's some T-dependent and T-independent responses that may be occurring. We also looked at this subset of donors that still produced flu-specific antibodies, even under CD4 depleted conditions, to understand whether they were equivalent in quality compared to the wild-type control cultures. What we did with bilayer interferometry is look for the dissociation rate, which is roughly a proxy for the affinity of the antibodies. You can see that under CD4 depleted conditions, the dissociation rate is increased, suggesting that those are poorer quality antibodies compared to the wild type cultures. And that was true for whether we looked at the full length or the head specific antibodies against A. California in particular. And a more recent question that we've been trying to address is whether memory CD4 T cells or naive CD4 T cells provide better help to naive B cells. So under these minimal type conditions, where the only B cells that are capable of responding are the naive low affinity ones, we wanted to understand whether CD4 T cells were providing a selective pressure or were generically helpful, and whether that was dependent on memory or naive phenotype. So we looked at this with 5 different donors. We looked at both the plasmablast differentiation potential of those cultures and the flu-specific antibody response.
Lisa, how did you isolate the memory CD4 cells? Is this flow using specific memory cell markers? Or?
Yeah, so this is all with sorting, and generally we would use CD45RA and CD27 to isolate naive versus non-naive cells. So when we look at these individuals, it appears, for the most part, other than this one donor, that actually it's the naive CD4 T cells that were more permissive to B cell differentiation into plasmablast phenotype. The interesting thing about this individual is when we go and look at the antibody response, they produced absolutely 0, which suggests to me that these plasmablasts were not actually responding to the flu vaccine stimulation we provided, but potentially to just something else that happened to be ongoing already in the cultures. So overall, it actually seems like the naive cells were perhaps better, or at least non-inferior to the naive, to the memory CD4 T cells in this case. And you can imagine that there might be several reasons for this, but my hypothesis is that the memory cells are more selective about which cells they're allowing to differentiate into plasmablasts, whereas the naive cells may just be pumping out generically some cytokines that will help sustain B cell differentiation. So what I've shown you is that the tonsil organoid cultures are capable of producing this multifactorial response to flu antigens, and that this is happening with roughly physiologically relevant kinetics. So I didn't have time to show you, but we've also looked, for example, for flu M1 specific CD8 T cells. We see that those expanded as well. And so we have a good idea that we have both T cell and B cell responses ongoing here. Through imaging studies, some depletion studies, and also repertoire sequencing, we can show that there's a maturation of some pre-existing immune responses, and also that the B cell response at least is capable of producing some new B cells. We've also done depletion and reconstitution studies to help understand new biology about these adaptive immune responses. So I'll just point out a few of the advantages that I've found compared to previous systems that I've worked with. The first is that this is a fully human system. So we're not using any cell lines, we're not producing any recombinant protein production, things like that, to be able to get these cultures to go. We just supplement them with a little bit of human BAF, and that's not even required. It really just helps the cell compositions over time. We can look at important cell types that are not found in the blood, so things like germinal center B cells and T follicular helper cells, which, unless you get just the right time point, are going to be very difficult to look at in the periphery. And also we've been able to scale this to a high throughput and reproducible and cost-effective technique. So we can actually scale all of this down to a 96 well transwell plate with very few issues. So I'm really excited about the potential of this to do more mechanistic experiments than has been typically possible with human samples. So I'll finish up with a few little pieces of new data that we can talk about, and also what my plans are for the future. So recently I've shown that the organoids can also respond to other vaccines. In this case, we're looking at the measles, mumps, rubella vaccine, or MMR vaccine. You can see in unstimulated versus MMR-stimulated cultures that we see increase in most donors in plasmablast production. And largely, most of the response actually seems to be driven against the measles component of the vaccine. There are clearly a few donors who are still making a decent mumps response and a little bit rubella. But this also is consistent with what we would see in vivo, where the measles response seems to dominate. We've also been
Lisa, I
able to
wonder if that's related to the, you know, to the resurgence of measles. I mean, depending on where you get your donors from, if you get them from the anti-vax community, then that's probably
So
explains it.
we do ask them about their vaccination history, and I would say almost every single patient says they're up to date on their vaccines. So we're taking their word for it, but you know, there's a possibility that there's circulating measles in the population. It's not impossible. So right now we're trying to get, we're grappling with the naive response, because clearly that's going to be important for future vaccine development. And one way I've been trying to get at this is by using essentially a foreign antigen that is fluorescent for us to be able to report and detect on the B cells that may be specific for it. So this is used as a trick in the mouse literature too, but people use phycoerythrin. Most of you are probably familiar as the label for a lot of antibodies for fax. It's actually a decent antigen in mice at least. And so we've been using that as an antigen, and then we can then restain cultures with PE to look for fluorescently detected B cells. And essentially, we set up these cultures, stimulate them with the antigen of interest, either no stim, an irrelevant stimulation, or with phycoerythrin itself. And you can see that the percentage of PE-specific B cells is about 10-fold increase when we stimulate with PE. And that this is not a generic effect because it doesn't happen when we stimulate with flu vaccine. We can also try modulating this response with adjuvants. In this case, we're using a TLR7 stim, imiquimod, and also just an alum hydrogel. And in this particular case, we only saw an increase in PE-specific B cells when we added alum to the cultures. Another question that I get a lot is whether this could be translated to other lymphoid tissues. So Anne Sperling at UChicago kind enough to send me a few samples from her her cohort of lung draining lymph node samples and spleen samples from humans who are donating their organs. And we found overall that the system works quite nicely with both lung lymph nodes and spleen. And just from personal experience, I can tell you that the cultures look really interesting too. So for example, the spleen samples look very spherical in their clustering, and the lung lymph nodes look kind of lacier, much more like the tonsils do. Overall, we do see when we stimulate with live attenuated flu vaccine or with actual A California flu virus that we see generally increases in plasmablasts. And we also see not so much CD4 T cell stimulation, but we do see some CD8 T cell stimulation. And importantly, we do see flu-specific antibodies being produced both against the vaccine in lung lymph nodes and in spleen, but also against the A California virus. So that would be cool in that it will allow us to look at even more elegant structural features, perhaps from things like spleen and lymph node compared to a more simple tonsil type structure. We've also used these same samples to look at naive responses, and we do see some evidence with the naive vaccine, in this case rabies vaccine, that we can see some small hints of an IgM response, but we don't see an IgG response from these cultures. So overall, when we look for rabies-specific antibodies, clearly there is not much going on in the IgG rabies-specific response, but we see consistently a little hint of an increase in all of the rabies simulated cultures compared to the unstimulated control cultures. So future work. I clearly see a future for understanding how antigen seniority works for flu immunity, which I'll discuss in a second. and also for testing in a high throughput way different vaccine candidates and adjuvants that perhaps require something more sophisticated than what we would typically see from your classical inactivated vaccine response. So something that requires sort of a coordinated T cell and B cell response. And also, I think this is a good way for us to better understand immune modulation because we have more control over the cell differentiation processes, potentially the tissue microenvironment. We could manipulate things like the extracellular matrix or even gene expression, uh, which we can provide transiently. So, to the idea of antigen seniority in influenza, some of you might be aware of this. It's also known as original antigenic sin in the flu literature. But basically, there's this idea that whatever your first infection was as a baby, that strain somehow affects your ability to respond to all subsequent flu infections in your future. So there's a lot to be done here because that evidence is largely from epidemiological studies. And you can imagine now that there are improved vaccination programs for young children, especially in this area, that it would be very interesting to understand how an exposure as your very first response to flu would be affected if it was a wild type virus that you actually were infected with, or if it was the inactivated vaccine or even a live attenuated vaccine. And this could vary quite a bit because we're talking about a response to a single strain versus a quadrivalent response to the vaccines. So I've just started collecting some preliminary data on this by looking at plasma from the same children's cohort that I've been looking at with the tonsil organoids. So here we're looking at blood plasma, just to get an idea for whether children have an H1 response pre-existing in their blood, an H3 response, or both, suggesting potentially either infection or vaccination, at least to start. You can see here, this is the H1 response. We see children even 2 years of age that have a California specific antibodies. And this particular individual also has an H3 specific response. So it could be that they were vaccinated as a 1-year-old. But that there's also variation. So we can see, one example of a 3-year-old here with no discernible response to either of these two strains. We can see people with just individual responses as well. So I think we can learn something by understanding these differences of pre-existing exposure, and then what the effect that might be on the organoid cultures, either with single strains to these individual groups or in the context of a vaccination. Of course, I'm sure this is on everybody's mind. Can we use organoid systems like this to study coronavirus infection, immune responses, and potentially interventions like vaccines? So I think it should be possible, and this is what I would propose for testing that. We already know, based on some literature that's just coming out now, that the ACE2 protein, which is the receptor for coronavirus binding, seems to actually not be highly expressed other than in intestine and in a few other tissues. But we can actually use the tonsil as a physiologically relevant antigen uptake system because it has M cell-like cells like you would find in the intestine, which are kind of non-specifically sampling the external environment. So how we propose this is a little bit different than the flu system that I've done before, but we have used this with other flu antigens, which is we can take total tonsil tissues, dissect them into a few pieces, and then actually use the epithelial interface to directly contact with either coronavirus itself, coronavirus proteins, or even vaccine candidates, and thus encourage antigen uptake in a physiologically relevant way. We can then mechanically dissociate the tissue into a single cell suspension and produce organoid cultures from those as well. So I think this could be a really excellent way for us to better understand the requirements for the expression of the receptor and viral entry. So as we were talking about recently, actually, it appears that interferons may be important for enhancing the receptor expression, and we could potentially treat tissue cultures with such things to be able to look at whether that affects the quality of the receptor expression. We can then look at the downstream immune response to coronavirus antigens. And we can look at this in a holistic way, right? Because there's both B cells, T cells, antibodies, and other immune cell types all together in the same culture dish. And I think this will be really important for being able to understand why is there variance among individuals in the types of response that they produce. And we already know that there seems to be substantial differences in young versus old people in the types of responses that are being produced and the symptoms that they get after coronavirus. And of course, I would be very excited to test vaccine candidates in combination with different adjuvants to see if we get something that looks like it would be an effective treatment. And potentially even screening individual B cell clones for the ability to produce neutralizing antibodies against coronavirus. And you can imagine that could be used as a way of producing a large amount of monoclonal antibody for providing passive immunity to people who are perhaps at high risk. So I will stop there. Of course, I have to thank my mentor, Mark Davis, for letting me work on all of these crazy experiments, and all the people in the lab who helped collecting tissues and processing them and doing various technologies like sequencing and bilayer interferometry. My collaborators in the organoid side, Calvin Kuo and Amin Salahuddin. My repertoire sequencing collaborators, Peter Kim and his grad student Mike Lyons helped us with acquiring different protein antigens to try, as well as all of our collaborators at Stanford and elsewhere who provided us with various antigens to test. For now, if you have questions, feel free to contact me at my Stanford email address. Hopefully, eventually you'll be able to find me in the UCI catalog, but I'd be happy to take questions now.
We haven't done this. Like, unmute, show yourselves, and give this lady a round of applause. That was great, Lisa. Really interesting. And what you didn't mention is that you have a Nature Medicine paper that got good reviews, and you're about to, any minute now, any day now, any minute now, about to resubmit that.
Yes, I'm trying to finish every possible experiment I can do in the next 40 days before I hopefully move.
Yeah, but you're only allowed into the lab for 2 hours a week or something, is that it? Under lockdown?
As long as I can stick some coronavirus stuff in there, hopefully I can address it.
All righty, that's great. Well, I have a number of we have a number of questions from the chat group, and people can feel free to ask. Well, I'll ask the ones that are up here, but please feel free at this point to unmute and, as I say, show yourselves and ask questions directly. But I'll start with the first one that was early on in your talk that one of our grad students, Will Pandori put in, and he asks, how long can the organoids be cultured? How many organoids can you make from a single donor?
Yeah, so we've gone out for up to 23 days. There's certainly viability loss over that time, but the cells that have already differentiated, especially plasmablasts, seem to hang around. So you can imagine that there's just this continuous selection process where all that you're left with at the very end, once all the antigen is gone and the antibody's been produced, is effectively the few remaining plasmablasts that were relevant and long-lived. And then in terms of how many cultures we can make from an individual, we get roughly 3 to 6 billion cells per tonsil pair. So that's if you're getting whole excisional tonsillectomy. So that is a lot of organoids. We can make something like 500 to 1,000 organoids.
That's really impressive. 1,000 organoids from one tonsil.
Yes, it takes a long time, I can tell you. I've made about 400 at once.
Wow. All right, so what you need is some good grad students in there.
That's right.
So Francesco was next. He has 2 or 3.
Eric.
Yeah, you can go ahead and ask them yourself, Francesco. You don't need
one of the questions that I have, I wrote so many, I apologize for that. But it is strictly closely related to what Will just asked. And it is the following. So do you envisage there is a life, so to speak, for B cells, or for what happens in germinal centers or in your organoid cultures, of course, after the organoid cultures are done? In other words, can you use the organoid cultures to, as a sort of a screening platform to get very cool B cells that do excellent antibodies and take the opportunity of, you know, sequencing those sequences, their BCRs, and basically take things from there beyond the organoid culture?
Yeah, so let's start with the in vivo situation. So during the germinal center response, the vast majority of B cells that get recruited die, right? That's the life and death of a B cell. But there will, of course, be those few that make it through and get selected. And so I think to at least that degree, the fact that there are cells dying doesn't really concern me for the organoids. That's just a part of the process. But I do think that it's a great way for us to be able to then go back and pick out the cells that we do think are relevant by just waiting, right? So yeah, I think it's certainly possible we could then single cell sort the B cells that we think are interesting, grow them up a bit, and then actually look for their antibodies with, you know, affinity measurements and things like that.
And did you want to ask any of your other questions here, Francesco? And we have some time. We don't have a lot of time. We have some time.
No, go ahead, man.
That's good. Okay, so actually, Will had another question. Well, why don't you wave your hand so Lisa knows who you are, since you're right there. Do you want to just ask it, Will?
Okay. Sorry. Okay. Yeah, I was just wondering, in your future directions, you touched on that you wanted to do this, but I wanted to hear a little bit more of your plan on how you're actually planning to test your vaccine candidates and adjuvants in your system, and and how you're planning to test your hypothesis that this system can predict efficacy of vaccines, at least as well as mouse models. And so I was wondering if you already had any targets in mind or more about what you're planning.
Sure. So I do have some preliminary data on testing some relevant and some, you know, in vivo only type adjuvant systems. So you can imagine we can supplement with things like different cytokines that you probably wouldn't put into a person, but could have physiological relevance. We found an appropriate target. And we've done that with things like the inactivated flu vaccine for now, and compared that to the commercially available flu ad, which is an MF59 adjuvanted flu vaccine, mostly for elderly people. And we can certainly see that we can provide some adjuvant type signals in combination with the inactivated flu vaccine that do as good as the commercially available adjuvanted vaccine. And they're not necessarily stimulating the same pathways. So I think that's at least giving us some preliminary data that would suggest we can use this as a system to at least test some adjuvants. Certainly it's not going to work great for things that supposedly make antigen depots or just recruit cells generically to, say, the muscle tissue for an inactivated vaccine. But we can look at things that may have cell intrinsic responses.
This is Phil. Lisa, I know that it's kind of a follow-up of Francesco's questions about looking more carefully at each individual B cell. I remember when you were here last time, you were still working on getting the sequencing of the individual B cells. And I see you have Bill Robinson on here now.
Yes.
So I guess, and you mentioned repertoire sequencing in your talk. So how's that going now? Is that, can you get a whole repertoire now and look at that map?
Yeah, so the circular dendrogram that I showed is the single cell sequencing data.
Mm-hmm.
And effectively, you can imagine the next step, if you wanted to just make monoclonals from that, would just to be go and get those synthesized, right?
Yeah.
So we can certainly do that. The problem with the single cell BCR sequencing is that you're largely restricted to the B cells that have surface expression of their IG still. So we still don't have a great way of sequencing fixed cells where we may need to do intracellular staining, because as you know, plasmablasts reduce their surface IG expression over time.
Mm-hmm.
Yes, so we're probably missing out on the best B cells there. Really the best way to do this would just be build up a repertoire of known specific sequences and then just sequence all of the B cells and look for the ones that have similarities and have those synthesized and look for those antibody affinities.
After you permeabilize the cells, is it hard to get the sequencing data from those individual cells?
Yes, so we have never been able to get that to work, especially from single cell, but we're using an RNA method. And so it could be workable with potentially a DNA method like Adaptive Bio uses.
Mm-hmm. Okay.
Yeah.
Lisa, I have a quick question. This is Craig Walsh.
Hi.
My question is related to something that you showed, which is, most of this has been focused on tonsils, but you also showed spleen samples and so forth, which of course are much more invasive to get those. I think those were coming from cadavers or something like that. So the question is, how reliable do you think using the tonsil as a sort of a system to recapitulate what's happening in vivo, and in particular, that's relevant to the site of where an immune response is normally happening? Let's say you have an anti-tumor response or an autoimmune response that's happening in a very different place in the body. I'm just curious about whether you think that the tonsil system actually will recapitulate some of those tissue-specific immune responses.
Lisa, why don't you stop sharing so we can see you?
Sure.
Or we can see Rebecca.
So I think that's an excellent question. I think we're not going to know until we try. So of course, I'm happy to produce organoids from just about any tissue with lymphoid cells and see what happens, but we're just going to have to try it first. But I do think we can still use the tonsil system as sort of a generic way to understand the cellular interactions that may be occurring and how we can, maybe develop some hypotheses that we could then go test in either an animal model or with, primary tissue samples.
So there's another question here from Roberto Tinoco, who is not showing his face there, but that's okay. He's showing a picture of his face. But Roberto, do you want to ask this? Okay, I'll ask it. He said he wants to know if the antibodies that you generated, the anti-flu antibodies, were those actually neutralizing antibodies?
They are. So we tested with a pseudovirus neutralization assay. And we just tried with a couple of the strains in the vaccine, but they were capable of neutralizing one of the B strains and the A California H1 strain as well.
That's great. It's very pertinent for flu, obviously, but also for COVID-19.
Mm-hmm.
Any other questions, thoughts? That's all the chat questions I have. Okay, otherwise, this is not bad. We had 34 people tuning in here. Lisa, of course, you were one of them, but that's okay. 33 is not bad. I would say another round of applause and thank you. And we're going to look forward to having you here. And I'll talk to you whenever, Lisa, we're around.
Please don't hesitate to reach out. I'd be delighted to hear from anybody. And I look
Our
forward to
info
meeting.
is on the IFI website. All right. Oh, look at that. You're getting
Thank you.
Emoji applause. All right. Thanks. Okay. Take care. We may talk about doing this in the future. It seems like a good forum by Zoom, so we might talk about that and maybe do something even more informal like Chalk Talks as we go on. But anyway, thanks all for attending, and thank you, Lisa. Take care for now. Rebecca, you can end this for everyone, and I'll talk to you soon too. All right. Take care. Bye-bye now. Bye, Craig. Bye, Rebecca.
Bye, Eric.
Bye, Rebecca. Thanks for doing this. I think we'll try to
Alright.
get something else going with this. Alrighty.
Okay.
I have one. All right. Take care. Talk to you soon. Bye.
Okay.
I
Bye.
got more meetings to go to.
Of
Never
course,
end.
all day long, right?
God, tell you. All right. Talk to you soon.